Hi everyone, and welcome to this short little lecture on ancient DNA and how we can use ancient DNA for exploring our past. My name is Linus Girdland, and I'm a lecturer in biomolecular archaeology at the University of Aberdeen. I just started, I took up my role here about 6 months ago. Earlier in 2020, and in this time I've established we just started two MSc programs in biomolecular archaeology and bioarchaeological science, and we're hoping to expand these programs over the coming years. Let me see here if I can change the slides. Yes, so I thought I'd introduce myself a little bit more and my background. I started my career really in archaeology, and I studied traditional archaeology. I participated in archaeological digs. Then I found osteology to be quite interesting. So I studied osteology, and my first degree is actually in archaeosteology. So looking at animal bones and human bones from archaeological excavations. And there's quite a lot we can say by analyzing bones. However, I found the emerging field of ancient DNA to be particularly interesting, and that's what I decided to pursue my PhD in. And so I moved to England. I studied first in Sweden, where I hold my first degree. Then I moved to Durham University to study pig domestication using ancient DNA. And this was about 10-15 years ago, when the methods that we use for studying ancient DNA were pretty basic. This is what we look like in when we dress up and study ancient DNA molecules in a laboratory setting. And the reason for that is that any DNA that is preserved in an ancient skeleton like this is so fragmented and damaged and present in so few copies that even tiny amounts of modern contamination, so DNA that I would deposit onto a bone if I were to hold it, for example, can outcompete the ancient DNA molecules present in an ancient skeleton. I'm also quite lucky in that I have good friends and colleagues who are active archaeologists, and so they go out to excavate. And so I'm now working with Veronique Forbes and Paul Ledger, who completed their PhDs at the University of Aberdeen just a few years ago. They work now in at Memorial University in Newfoundland, where they excavate, in this particular case, Lance aux Meadows, which is the only confirmed Viking settlement site in North America. And I was lucky enough to be able to go out there, and they have now found funding for a big project in which ancient DNA will form a small part. So not only can we extract DNA from an ancient skeleton like this, but what we are doing in this particular project is to extract DNA from sediments in naturally accumulated peat immediately surrounding the Viking Age or Norse settlement. So that's an interesting project that I'm looking forward to working more on. So the field of ancient DNA, as I mentioned previously, just 10-15 years ago when I started this career path, the methods were fairly basic, and we had difficulties obtaining ancient DNA. But over the past 10 years or so, there's been a massive increase in the number of ancient genomes we have available, and this is because of optimized methods and a lot of research going into exploring really how we can best extract DNA from these ancient skeletons and how we best analyze them, right? So over the past 10 years or so, there's been a rapid increase in the number of ancient genomes that have been published. And not only can we extract or analyze small parts of DNA, we can now analyze whole genomes essentially, and by doing so we can extract the total ancestry information held in ancient genomes. So some scientists have even exclaimed the ancient DNA revolution. And it is within this context we're trying to establish new masses programs and integrate with the broader field of archaeology. Let me see here. So just last week in Science, apart from Science is a big science publisher, essentially. The Science is the name of a scientific journal, right? It's one of the more prominent ones, I would say. And in last week's or last month's version of this. We found 3 ancient DNA papers amongst a bunch of COVID-19 research and so on. So ancient DNA is very much at the forefront and the cutting edge of different types of fields of research. And in this science particular journal, in this edition, we found ancient DNA research that resolved the questions about ancient dogs and where and when dogs were first domesticated. So extracting DNA and analyzing DNA from ancient dog bones. And we found a research paper looking at the paleogenomics, so whole genome data of denisovans, so cousin species of the anatomically modern humans in late Pleistocene, so very old Stone Age sediments from a cave site in Tibet. And following on from that, they placed this in a broader human evolutionary context and explored how denisovans, so this cousin species of modern humans, contributed genetic ancestry to modern East Asians. So a whole range of different research questions that are becoming accessible because we can now sequence ancient DNA molecules, and much better so than we could just 10 years ago. I will tell you a little bit about 3 case studies and highlighting some research that I'm working on and the contributions that we have made for the broader understanding of human prehistory. And we can look at this case study first. It's concerning the Aboriginal population and original inhabitants of the Canary Islands, the Guanches. And their origin is still not fully understood. It's hypothesized that they were placed on these islands, for example, by Romans a couple of thousand years ago. So when Spaniards invaded these islands in the 1480s, I believe it was, in mid-1400s, they found a group of people that we know of as Guanches on each of the 7 main islands in the Canary Islands. Yeah? And it's, for those of you who don't know where that is located, this is off the west coast here of North Africa. What was interesting was that, and why the origins of this group of people is particularly interesting, is that when the Spaniards invaded these islands, they were met by a people who lived mainly using Stone Age technology. So they relied on goat herding, they did not have metal, and they could not sail. So there was no inter-island communication here, right? So it's a bit of an enigma, and that's why their genetic origins could potentially shed light on this. So in this particular study, we obtained bone samples from 12 Guanche remains, right? And we were lucky enough that the Anatomical Museum in Edinburgh, which houses a great collection of anthropological remains from around the world, which of course is linked potentially to some ethical issues. The museum is very much partaking in repatriation programs and things like that. However, because the Guanches have left no living relatives, in the sense that there are modern groups associating or identifying themselves as Guanches, we could ethically sample these as we do with other archaeological remains. And we extracted DNA from teeth or from other bones in these skulls, and we radiocarbonated some of these. So we extract proteins, and we can measure the relative presence, shall we say, of different isotopes, right? Carbon-14, and we could directly then, well, we can estimate the precise age of these specimens, and they were very much forming a nice time transect here. It's hypothesized that the Guanches first arrived on the Canary Islands around the time of Christ, right? So about 2,000 years ago. And the Europeans and the Spanish invasion took place in the 1400s. And the samples that we extracted DNA from form this time transect across this period. And we managed to sample individuals from 2 of the islands, so from Tenerife and Gran Canaria. So there is some spread to the geographical location of these individuals. And what we found is that they group primarily closest to modern day North Africans, which is not that surprising. It's in fact, it's been hypothesized, right? But it was nice to be able to confirm that using ancient DNA. And we looked at 3 different types of genetic markers that are informative for inferring the geographic origins of a group of people like this. So we looked at the Y chromosome, which is inherited solely on the paternal line. So it's a lineage marker. And we analyzed mitochondrial DNA, which again is a lineage marker, but that captures that passed on, inherited on the maternal line. So it captures the different line of ancestry, essentially. And this figure here depicts the autosomal ancestry, which captures the genetic ancestry from all of our genetic ancestors, essentially. It's not a lineage marker per se, but because of processes such as recombination during meiosis and during the formation of gametes, chromosomes are shuffled up in between each generation. So genetic ancestry from several ancestors is captured within the autosomal genome of any given individual. And it's by accessing this type of genetic information that we can place the guanches, in this case, within the broader genetic landscape of humans. And so we assembled not only a reference population comprising modern North Africans, particularly of Berber origins, so Moabites, Algerians and Tunisians, but also Egyptians and modern Saudi Arabians and Bedouins, Palestinians. And then moving into Europe, we assembled reference populations comprising then Italians and Greeks and Sardinians. And simply by accessing this autosomal genetic variation within the guanche skeletons that we sampled, we could place them within the broader genetic diversity of modern populations. And we can confirm then that they show very close affinity to modern North Africans, thereby informing archaeological research where to look for their origins in case we want to investigate that further. And that was further followed up with different types of analyses of this autosomal genome or autosomal genetic ancestry that allows us to quantify relative proportions of different deep genetic ancestries, right? And we found some evidence of sub-Saharan African genetic ancestry, some Near Eastern genetic ancestry, but primarily this green color here, which is represented by modern North Africans. We could then move on, and this is what we did. So I mentioned earlier that there are no living relatives, and that's actually a false statement. There are living relatives, and we could show that by analyzing the genomes of modern Canary Islanders. We found traces of guanche ancestry in modern Canary Islanders to the exclusion of other European groups. So the majority of people living and who can trace their ancestry back several generations in Canary Islanders in the Canary Islands are of Spanish ancestry, right? But we found evidence that up to 16 to 30% of the genetic ancestry in modern Canary Islanders derive from the guanches. And it's a pretty crude estimate, but we only have access to a couple of good genomes, but it's indicative of genetic continuity. What is interesting is that we found no continuity in the Y chromosomal ancestry. So if we look at this male lineage marker, we found no evidence that modern Canary Islanders are carrying essentially guanche Y chromosomes. So that allows us to hypothesize that the Spaniards, when they invaded the Canary Islands in the 1400s, killed off most of the males, because we know there were wars waged on these different islands when the Spaniards arrived, right? And so the surviving guanches were probably primarily females. So and they later became assimilated within the Spanish population. Lastly, we can also access information about the phenotypes, right? So skin color, hair color, and things like that. And there are a few mummies that are preserved and in display in various museums in the Canary Islands primarily. And so we could hypothesize that the guanches carried, for example, dark hair and brown eyes and things like that. And we sequenced one genome in particular to very good coverage, allowing us to look at genetic information contained in the genes in the genome, right? So genes are sequences of DNA or regions of chromosomes that encode for proteins, and that will then directly determine the phenotype of an individual. And we made use of a forensic reference panel of genetic variants that would allow us to estimate the eye and hair color, the skin pigmentation, and various other genetic markers linked to eye and hair color in this particular case. The University of Dundee and Sue Black and her colleagues have moved on or continued this research and made a facial reconstruction of one of the iguana skeletons held in the Anatomical Museum. So we've gone essentially from just a skull that was sat in a what is essentially a Victorian collection, anthropological collection, to being able to place these individuals within the broader landscape of genetic variation, thereby informing archaeological theory about where this group of people originated, to being able to reconstruct a face like this using, in part, some of the genetic information we extracted. And importantly, we were able to demonstrate some genetic continuity from these guanches into modern living populations. So that's quite a travel from just being sat in a collection like this, and all of this was achieved just by extracting some ancient DNA. Some other research we are currently doing is we're performing similar analyses on the Picts of Scotland, and this is in collaboration with other colleagues in the archaeology department here at Aberdeen, so Gordon Noble and Kate Britton, who of course have very well established research programs on Picts. But of course, we by adding this biological dimension to the analyses of, or to our interpretation of the Picts, we hope to be able to broaden the overall narrative and understanding of who these guys were, because in all honesty, there's quite a lot of question marks as to their origins and genetic affinity with other populations. There are plenty of hypotheses out there, but by accessing DNA from Pictish individuals, from Pictish burials, we can directly access, we can directly test these hypotheses. And what we find when we compare Pictish genomes, so here we have 1, 2, 3, 4. The circles here are Pictish genomes. Again, compared over genetic variation of modern Europeans in this case. So here we have, Northwestern Europeans, Southwestern Europeans, and so on. And what we find is that the Picts fall into the landscape, shall we say, the area of this particular plot where other individuals from Bronze Age and Iron Age UK or British Isles cluster, right? And so what happens in Europe towards the late Bronze Age, towards the early Iron Age, is that all these different deep ancestries that we find, for example, in Neolithic farmers, so Stone Age farmers, or even older Stone Age individuals from Mesolithic Britain or from Scandinavia, all of these ancestries combine into what is this modern European variation. So modern European variation essentially forms towards the Iron Age, and this is also reflected in the genomes of Picts and other Iron Age individuals. Of course, this is preliminary research, and we are continuing our efforts to resolve the finer genetic relationships amongst the Picts, or so within the Pictish communities and within the broader European contexts. Lastly, I would like to highlight a case study that we published just a couple of years ago, and in which we looked at the potential kinship in individuals that have been buried in megalithic tombs. And the megalithic tombs is again a kind of late Stone Age phenomena that is concentrated primarily along the Atlantic facade of Western Europe, and it's particularly prominent in the British Isles. You will all know about megalithic tombs in Orkney and in Ireland and so on. And so by extracting DNA directly from individuals buried in these tombs, we could look for kinship, so very close relationship amongst individuals. And this particular plot demonstrates some of these kin relations that we observed, specifically in Carrowmore, which is a big megalithic tomb in Ireland. We found several kin relations across generations. because again, we can use radiocarbon dating to place these individuals on a temporal sequence, and then we can look for genetic relatedness across time. And we found several first and second degree kin relations in that tomb, suggesting that megalithic tombs were linked to kinship. And this was later confirmed also in a few cases for megalithic contexts in Scandinavia. So that's the kind of northeasternmost extreme of this archaeological culture as it spread along the Atlantic facade. There's been follow-up studies, and just earlier this year, an Irish research group published more genomes, and they found additional evidence of that kin relations and kin structure is linked to the formation of these megalithic tombs. And on that note, I open up this session for some questions. Thank you. In our undergrad program, so we have essentially a standard archaeology program. It comes in the MA version or a BSc version. We provide a very broad background, both to traditional archaeology, gaining getting skills in excavations and those traditional types of methods. But we also include lectures and seminars and practicals on each of the for each year, essentially, for different courses and different modules where we deal with things like ancient biomolecules, right? So you'll be introduced to the study of different ancient biomolecules within the undergrad programs. And so this leads to, this allows you to really continue with ancient biomolecules in case you're interested in this particular research field, right? And we do offer the biomolecular archaeology, so an MSc in biomolecular archaeology, and that is run based in archaeology, but in that particular program, you'll take courses over in biological sciences and biomedical sciences. So more kind of genetics and DNA sequencing and those types of aspects. The other program we offer, which places emphasis on ancient biomolecules, is bioarchaeological science. But in bioarchaeological science, you have the opportunity to take other archaeology courses and explore the context of archaeology in a more in depth. Okay, so ancient DNA is really an intersection between evolutionary biology, microbiology, biomolecular archaeology, and traditional anthropology, essentially, right? And depending on your interests, you can find a pathway through our different programs that kind of suits your interests. So we have plenty of offers, or plenty of opportunities for exploring these interests. And we have a question here. How did you have to go about obtaining modern Canary Islander DNA to compare to the ancient DNA? Actually, we did not do that. That reference data was already published in a population data set. I don't know precisely from which paper. There's a couple of different big reference data sets that have been published over the past 5 or 10 years. And so they contain population samples from across the world against which we can compare unknown samples, such as the Guanches were by the time we started to investigate them. So the way these reference populations are constructed is usually that the researchers go out and ask questions. Well, first of all, it's based on volunteering, right? So the researchers propose, I guess, to different modern Canary Islanders or British people, whatever. Hey, do you want to partake in this big genetic study? And can you trace your grandparents or can you trace your ancestry back 3-4 generations to the location you currently live? And if the answer is yes, then they would be suitable for being a data point, I guess, in that reference panel. And we have another question here. How is DNA incorporated into teaching in archaeology at the university? Okay, so I kind of hinted at that previously. The way we do it is for ancient DNA, in particular for the archaeology programs, is we provide kind of an introduction to ancient DNA in level 2, and I'm not sure entirely what the ancient DNA component is in level 3, but in level 4 you have a specific course called bioarchaeology, in which we have a mini module where we explore DNA inheritance, properties of ancient DNA, how we study ancient DNA in lectures and seminars. And we plan, because I'm fairly new to the university, right? I've only been here 6 months. We plan to incorporate more aspects of ancient biomolecules into undergrad programs. And of course, you can explore this in your research dissertation if you want. So the research project you should complete in your last year of undergraduate study, and that would of course set you up for continuing that in one of our master's programs. It's a question from Dan as to whether because of the dating of their arrival. Yeah, it's true. They possibly arrived earlier, so and we don't know precisely when the group of Aboriginals or group of people that we now label as Guanches arrived on the island. So there's some tentative archaeological evidence that the islands were settled perhaps much earlier, although the earliest directly dated human remains, I believe, is something like they're probably not older than 2,000 years. So there's a bit of question mark there as to population continuity within the Canary Islands. Okay, but it also depends on from where they arrived and from where they were sourced, if we use that term, right? So the source population that likely that the ancestral populations to the Guanches may not have been using metal. So anyway, Dan, to answer that last question, a possible case of cultural regression, I doubt it, to be honest. Possibly, but it's not my place to discuss that. I don't think so. We focus primarily on the genetics, right? And it's from our point of view, the cultural material will have to be kind of not necessarily reinterpreted, but the idea here is that the genetic data will inform the interpretation of the material culture and archaeological record in a more traditional sense for discussing these types of questions that you pose here. So those are excellent kind of follow-up questions for future research. So I'd like to emphasize that. So despite the narrative that is sometimes portrayed in the media, and the way we present this research is that, oh, we've solved this big issue and that's the end of it, and that's very much not the case. Again, it's worth emphasizing that the ancient DNA research that we produce and the data that we publish, it's more of a framework, right? It's the biological framework underpinning essentially material culture, because material culture is the expression of people, right? And people are biological beings, and so understanding the biological pathways or ancestral pathways by which humans have migrated and things like that. The idea here is that understanding that biological connection across different populations across time will allow us to better interpret the material culture. So and that's something that is, in my opinion, currently not achieved to a very great degree. So there is some research currently being formulated, I would guess, where traditional archaeologists looking at material culture are now more and more integrating with the ancient DNA side of things to achieve better understanding, I guess, of material cultures specifically. But it's still very much early days in that sense. And that's part of the reason why we established these master's programs within an archaeology department, and that is to better integrate the biological aspects within archaeology, really, as opposed to just leaving this side of research out, to population geneticists or biologists, really. Thank you, Dan, and feel free to email me if you have any questions. You can find my contact details on the Aberdeen website, of course. Okay, thank you all for attending, and it was nice to see that you are interested and had some well-informed questions, in fact. And I hope to see you at some point in the future, and feel free to contact me if you have any questions about our programs or about our research in general. I'm happy to answer any questions. Thank you.